Today I'm talking about DNA sequencing, and I'm going to be talking about sequencing coverage and sequencing depth. And to do that, I'm using a very simple fictitious genome here that is 4 blocks long. Let's say, just so we can put some numbers to this, that each of these blocks represents 100 base pairs of genomic information. Therefore, we are looking at a 400 base pair length of genome. Now, this is inside the cell. If I could get inside my tiny magic school bus and drive into the cell, I would see this genome. But I can't do that. As a scientist, I need to extract DNA from the cell and then sequence it with a DNA sequencer. And this DNA sequencer that I'm working with in this imaginary world can't sequence all 400 base pairs at the same time. It's only going to sequence them in the 100 base pair chunks. And this is true in real life too. So far, there's no sequencer that can sequence along the thousands and thousands and thousands of base pairs that would make an entire intact genome. We have to sequence it in chunks. So our task here is to get sequencing reads that will cover the entire length of the genome. And in here, by read, I mean that the sequencer would read along this portion of the genome and it would return back to me, the user of the sequencing instrument, the A's and the T's and the G's and the C's that made up this chunk of genome. But again, I'm not inside my magic school bus. This is all happening in a cell in a tube, and then eventually on a flow cell of a sequencing machine. So this is going to represent my cell and tube and sequencer, right? So here is my tube, and it has, in this perfect example, exactly 1 cell's worth of genome that has not been copied at all. We have exactly 1 little chunk of genome in there per section of the genome. So now you might be asking yourself, how many times do I have to reach into this tube or cell in order to get the entire genome? And now in this example, me reaching into the cell represents the sequencing machine reading that chunk of DNA. So you can pause the video or think about it for yourself. How many times do I have to reach in there to get the entire length of the genome back? And let's see. 1 time, didn't get the whole thing. 2 times, 3 times. I think you're realizing now it's going to be 4 times, right? There are 4 chunks to this genome, and now my tube is empty anyway. Like I said, in reality, we're never just looking at 1 cell, although single cell sequencing is getting a lot better. But even when we are sequencing a single cell, we still have to make lots and lots of copies of the genome in order for us to see it. So all of these blocks are representing those copies of the genome that we have to make in order to see it all, right? So I'm going to put them in here, turn it quietly. I know it's loud. Sorry. So here we have lots and lots of copies of the genome that I've made, and they're all mixed up in my cell and my tube. And again, the act of sequencing isn't informed. I can't say, oh, I want a red one. I want a green one. I have to just randomly reach into this bowl because that's what the sequencing machine is going to be doing. So now we have a question. How many times do I have to reach into that bowl to get the full length of those 4 different colored blocks out? In other words, how many reads do I have to get full genome coverage? And we're going to see why that is no longer just 4 in most cases, right? So here I am, I'm reaching in. Okay, got this bit. Got this bit. Ooh, I'm doing pretty good so far, actually. Uh-oh, so we already have a problem. I'm at 4 reads, but I still don't have a blue one. 5, 6, 7, 8. So in other words, I had to reach into the bowl 8 times. I had to get 8 reads in order to get at least 1x coverage of this entire genome. That is, I had to get 8 reads to see each section of this genome at least 1 time. Now, the number of blocks that I have stacked up here tells you the coverage at that particular locus. So you can see, or that chunk of the genome. So I had 1x coverage of this, 2x coverage here, 3x coverage here, and 2x coverage here. So on a genome-wide average, I had to get about 2x coverage in order to, or 2x sequencing depth in order to get full coverage of my genome. So just to refresh those concepts, we have two words that I'm using somewhat interchangeably. We do this, I'm sorry. But you can think of coverage as just, did you sequence the region at all? And depth, or depth of coverage, as we sometimes say too, is how many times did you sequence that region? And there's some math that we can do here. We calculate depth as the number of reads, in the example before, the number of times I reached into the bowl, the average read length, that is, on average, how long were each of the reads that I got when I reached into the bowl, and the genome size of the organism. So in our example below, or before, remember, I reached into the bowl 8 times. That is, I got 8 reads. I told you that each of those blocks, each of those chunks that the sequencer, which was me, was reading was about 100 base pairs long. And then the genome size was 4 chunks long, so 400 base pairs was our genome size. You can do some math here, and you can see where I got that number, that on average, our genome depth of coverage was 2x. But remember, you can go back and watch that bit before. I didn't have 2x coverage at every position along the genome. So that's why it's important here to know that we're talking about an average depth of coverage along the genome. In the last example, I was talking about sequencing coverage and sequencing depth just to see the sequence at all. But now let's say that we want to see variance at that sequence. So to mark these variants, I'm going to say that this individual that I am sequencing is heterozygous for a locus at each of these positions. So to mark that heterozygous locus, I'm just drawing marks, and I'm going to do them on both sides just to make it easy for myself. I'm drawing them on half of the 4 blocks to indicate that half of the loci in this individual have a certain allele. Let's say that's the, oh, I didn't do that side. That's the black mark that I just made, whereas the other half of the loci here do not. Now again, I'm sequencing this, so it's all going into my tube / cell down here. Now, there's a new question. How many times do I have to reach into the bowl, aka how many reads do I have to get in order to get full genome coverage and to know that this individual is heterozygous, right? I just told you that, but in a real world, you don't actually know that yet. You're trying to figure that out. So let's start reaching into the bowl, right? Reaching in 1 time, and I see our variant allele at that locus. 2 times. 1, 3 times. Trying to make sure these are all in frame here. 4 times. Ooh, I got full genome coverage fast. But right now, I can't tell that this individual is heterozygous at all of these loci. If I looked at this point, I would say that they have this one allele here, and they see different alleles at each position. So I have to get at least 2 to know that they're heterozygous, right? So here, ooh, on reach 5, I do it. I see that this individual is heterozygous at the red locus. Keep going. Oops. On reach 6, now to me, at this point, it looks like they are homozygous for not having that black mark, for not having that variant at the yellow locus, right? Which, as we know, is wrong. Ooh. Reach number, what are we on? 7. I do see that they're heterozygous here. 8. Oh, getting more blues. 9. Oh, I now see that they're heterozygous here. And I think we're already past the number of times that I had to reach into the bowl that first time, right? Just to get coverage. Now, I'm not only trying to get genome coverage, I'm trying to see what variants are there. So if you're keeping tally, it is more times so far, right? What are we at? 10, 11, I'm still not getting it. 12, I'm still not getting it. 13, lucky number 13 apparently. It's only after I got at least 2x coverage at every position, and you can see on average a lot more than 2x coverage at every position, that I can actually tell now that this individual was heterozygous at every locus that I'm looking at in this genome. And again here, by heterozygous, we just mean one of the copies has the mark and one does not, right? Now, you might be asking yourself, why does this matter? Why don't we just always sequence everything in the bowl? Well, first of all, you don't know what is in the bowl. And second of all, every time I reach into this bowl, that's us getting a read, and that costs money. So this would get really expensive, and which is not something that we want, especially if we're using genomic tools to, say, diagnose patients in a clinical setting, where we need to make these as affordable as possible in order to be equitable and for them to be a useful medical tool. All right, I hope that makes sense. Thanks.